welcome students to this YouTube class of HSS Pattern. This is ZAHOOR Ahmad and we are going to study today our topic, the structure of flower, that is there in the syllabus of your botany subject. So first of all, let's try to understand what flower actually is. Flower is a reproductive part of plant. Sexual reproductive part of plant is flower. Those angiosperms where sexual reproduction takes place, that sexual reproduction takes place with the help of this flower. So the first thing that we must know about the flower is that it is the organ of, it is the part of plant through which it reproduces sexually. Now let's try to understand what this flower looks like. What are different whorls it is made up of. As you are already aware that flower is made up of different whorls, the thing that we have already studied. Let us just have a recap of that. Flower is made up of calyx, corolla, androecium and gynoecium. Calyx and corolla, these two whorls are non-essential or accessory, but so far as these two whorls are concerned, these are of importance in sexual reproduction. These two whorls, calyx and corolla, though may help in sexual reproduction, but indirectly. But androecium and gynoecium are those parts of a flower which are directly involved in sexual reproduction. As we are already aware, this androecium consists of stamens and gynoecium consists of carpels. Carpels have other name, pistils. In today's topic, we are going to study about structure of anther in particular, because it is anther where the reproductive cells, the male gametes, are formed. So far as this anther is concerned, let's first of all try to understand what stamens are, then we will reach to the point where we will study structure of anther. Androecium is made up of stamens. As the structure is shown here, stamen has two parts. Its lower sterile portion is called filament. It is sterile part, therefore it does not take place in sexual reproduction. Microspores or pollen grains are not formed here. This is the fertile part of stamen and this part is called anther. This is called anther. You can see the longitudinal section of the stamen here. You will find it has got here filament towards lower side, which is sterile part of stamen, and towards its upper portion, there in bythecus anther there are two lobes, and each lobe has got two pollen sacs as shown here. So when there is bythecus anther, by saying bythecus anther we mean an anther which has got two lobes. There are also monothecus anthers. Monothecus anthers have got just single anther lobe which carries two pollen sacs or pollen chambers. Example is that of shoe flower, and shoe flower is there, cotton plant is there, where we find monothecus anthers. Here we have got bythecus anther, means it has got two lobes and every lobe has got two pollen sacs or pollen chambers. The thing that you are seeing here, this and this, these are two anther lobes. These are two pollen sacs on one side of this anther, and on this side there are two more pollen sacs or pollen chambers. So one lobe carries two pollen sacs and the other lobe carries two pollen sacs. It means there are total number of 4 pollen sacs or pollen chambers in this anther, which is bithekus. This portion, as you can see here, this portion of anther is also sterile, and it just connects these two parts together. So it is named connective. This connective connects this lobe with this lobe, and this connective has got in it vascular tissue. Vascular tissue, as we are aware, is the tissue which is concerned with conduction of water and minerals, and also transport of food. So this was longitudinal section of the stamen, which has this filament and two anther lobes. Now let's try to understand the transverse section of anther. I have made these diagrams on the board already to prevent the loss of time. Now let us try to understand what are the different anatomical features, what are the different tissues which make this anther. As far as transverse section of this anther is concerned, it is shown here on your whiteboard. The first layer that is present on the outside and which is common to all the 4 pollen sacs here. Let me first of all try to make you aware that this bilobed bithekus anther has got 4 pollen chambers or pollen sacs, which are first called microsporangia. These are first microsporangia in earlier development. Then after progressive development, a stage reaches when these microsporangia carry from microspore to pollen grains. Then the name changes. Before it is called microsporangia in earlier development. The stage when pollen grains are formed here, then its name changes into pollen chambers or pollen sacs. These are 4 in number in 4 corners of this anther. Now let us try to understand the structure of each microsporangium. This first layer of cells that surrounds all the 4 microsporangia, this is called epidermis. Epidermis of this anther is made up of cells which are thin, their cell wall is thin, and they are smaller in size. The stage when this anther matures, they shrivel. This is continuous layer which surrounds all the microsporangia and connect you together. These places where you find a sort of groove, these are called stomium. These are called stomium, singular stomium and plural stomia. They are actually the lines of dehiscence. When the flower dehisces, it sheds its pollens, it dehisces through these stomia. Then there is another layer of cells inside this epidermis, and this layer of cells is called endothecium. This is called endothecium. This endothecium is a layer of this microsporangium which is thicker in nature. It is thicker because its radial walls and inner walls are thicker due to deposition of cellulose in them. The stage, since this anther is in different stages of development, this develops up to the stage when it dehisces finally. In its earlier development, these are immature, and when the development is carried on, they are then converted into dead cells at the final stages of development. Now, beneath it, towards interior, there are 1 to 3 layers of middle layer cells. There is middle layer beneath endothecium, which is made up of 1 to 3 layers of cells, and this middle layer is made up of such cells which are smaller in size and they have thin cell walls. But in some cases they may also have thickenings. One thing that must be understood here is that in this region and this region, both epidermal cells and this endothelium cells, they are thin walled and small, because these are the regions to here has got, as I said, vascular bundles. And as I earlier mentioned, vascular bundles are meant for conduction of water, minerals and transport of food. This was about these three layers, I mean epidermis, endothelium and middle layer. These three wall layers are protective in function. Though Anther, microsporangium has four walls: epidermis, endothelium, middle layer and tapetum. But before going to discuss tapetum, let's first of all try to understand that these three wall layers of each microsporangium are protective in function. That means the prior importance of these layers is that they help to protect the underlying sporogenous tissue which is there in each anther lobe. Then comes the final wall layer, that is tapetum. This is tapetum. Tapetum is the inner wall layer that surrounds sporogenous tissue, and these tapetal cells are of utmost importance because of many reasons. Let's try to find out what are some important functions that is being carried out by cells of tapetum. The first important function of tapetal cells is that they are nutritive. Since this anther is undergoing development, by saying anther is undergoing development, we mean different parts of anther, especially sporogenous tissue, which is lying there in the innermost part of each microsporangium, is undergoing development. For the development of sporogenous tissue, there must be supply of nutrients, and that is being provided by tapetum. The cells of tapetum provide nourishment to this sporogenous tissue which is in the center. Then it has got one more important function, that it produces enzyme callase. Callase is an enzyme which dissolves callose which is there in the microspore tetrads. In the development of microspores in sporogenous tissue, first they are in the form of tetrads, and the tetrads are put together by callose. That callose wall is being dissolved by this callase enzyme which is being produced by tapetal cells. It also produces Ubish bodies. Tapetum produces Ubish bodies. Ubish bodies are the material for formation of sporopollen. Ubish bodies help in the formation of sporopollen, an important material which is responsible for the formation of exene and for its strength. They also produce, or I could say secrete, they also produce enzyme indole acetic acid. Indole acetic acid, as we must be aware, it is one of the auxins. This indole acetic acid is growth hormone. It helps in growth of pollen grains here in pollen chambers or microspores. Then it also produces compatibility. Incompatibility protein. tapetal cells produce protein. Incompatibility protein is that protein which helps these pollen grains to grow on certain specific stigmas and not to grow in certain specific stigmas. To recognize a particular stigma for the growth of pollen tube and germination of pollen grains, there is a reaction that is called incompatibility between stigma and between stigma and pollen grain. This incompatibility recognition is being supported by proteins. When there is compatibility protein in pollen grains and it has its association with stigma, then there can be germination of pollen grains on a particular this stigma. When there is incompatibility, the particular pollen grain fails to germinate on a particular stigma. These are some important functions that are being carried out by tapetum. So with this, we understood the function of tapetum and the different wall layers that are there in each microsporangium. Now let's try to understand something about sporogenous tissue. Sporogenous tissue lies in the inner cavity of each microsporangium, and this sporogenous tissue consists first of homogeneous mass of cells which are called sporogenous cells. First, each microsporangium is formed of homogeneous mass of sporogenous cells in its center. Then gradually, with the development of microsporangium, these sporogenous cells get converted into microspore mother cells or pollen mother cells. Pollen mother cells, up to pollen mother cells, all the cells here are diploid in nature, and we could say that all the divisions are mitotic in nature. One thing that we forgot, that we skipped when we were studying tapetum, the cells of tapetum are polyploid in nature due to endopolyploidy or endomitosis. Microspore mother cells or pollen mother cells, up to this stage, all the cells except those of tapetum are diploid. Now it is for the first time in life cycle that a plant cell undergoes meiosis. So pollen mother cells undergo meiosis and form microspores or pollen grains, we could say. Now in plant's life cycle, it undergoes change from diploid stage to haploid stage, and it is must for a plant to undergo this change because there needs to be fertilization. When there is fertilization, there is again restoration of diploid nature. Male sex cell which is formed here in pollen grains has to fuse with the female sex cell, that is egg, which is present in the ovaries in ovules. Now for that thing to take place, these have to change from diploid stage to haploid stage. When these change from diploid stage to haploid stage and form gametes, we could see in fertilization after their fusion, again diploid stage is restored in plant life cycle. So we could say the zygote, when formed after fertilization, reconverts plant stage from haploid to diploid, and up to the stage when it again forms pollen grains from pollen mother cells. This long period of plant is of diploid nature. The small period in the life cycle of plant when it is in haploid stage is that when pollen grains are formed, and that happens when pollen mother cells undergo meiosis. This was all about the topic structure of anther and microspore engine. Next time when we meet, we will study about microsporogenesis, how microspores are formed. Thank you.